Hey guys, just wanted to show my face just to assure you that I'm not just a computer program like Siri or something like that. I am an actual live person. For some of you guys, maybe I didn't meet you when I first came to Waxahachie Global a few weeks back or a month or so back. Maybe you didn't get the chance to say hello or see me there. So I know this first exam kind of kicked our butts a little bit. We didn't do very well in this first exam partially because we didn't have a textbook, and then I think partially because we weren't as well prepared as we should have been. I have allowed the retake, and I see where most people have done quite a bit better on this retake. That probably is because I left the first exam up with the answers available, so I know some people probably took advantage of that. So I want you to be assured that I think you will improve as time goes by. And then also I want you to keep in mind that this will be the only retake that we get this year because of the textbook situation and because of our first year starting out. We had some problems getting started. But as we go on further down the line, I would expect that we wouldn't have to have any more retakes, and I know that we'll be well prepared. So we're moving into Chapter 6. We're going to be talking about cells. We're finally up to actual living objects. We're no longer talking just about molecules, and I think things will go smooth from here. So good luck throughout the rest of the year, and let's get started on Chapter 6. Alright class, so here we are on Chapter 6. We're finally up to living objects. We're finally up to entire cells, which are the smallest unit that we would consider alive. So we're finally into what you probably thought biology was going to be all about, which would be living creatures. So let's go ahead and get started with a basic overview of our chapter here. So the overview says that the fundamental units of life, that's what a cell would be considered. All organisms are made up of cells. All living organisms are made up of cells. The cell is the simplest collection of matter that can be considered alive. In other words, it does all the things that we consider that living things do, such as reproduce, grow and change, adapt to the environment, things of that nature. Cell structure is correlated to cellular function. We keep seeing that over and over again, where form and function go hand in hand. All cells are related by their descent from earlier cells. If we take a look at all this information right here at the top, these 4 or 5 points all taken together make up this concept, this idea known as cell theory. So you may have heard of that before. It's one of the major scientific theories that we now have, called cell theory. And again, there's that word theory again. Hopefully we now understand that a scientific theory, what our deeper understanding or meaning of that word is, and that we understand that a scientific theory is an overall explanatory statement that is backed up by large amounts of facts and information, experimentation and documentation and evidence. So this theory is kind of an explanatory idea that could also allow us to make predictions and do future experiments regarding cells. Awesome pictures in the textbook in this particular chapter. Your textbook does a great job of showing all sorts of cool pictures, and particularly in this chapter I think it does a great job. Here is some brain cells, and you have in kind of the purple you have the branch of a nerve cell reaching out to the orange yellow other portion of another nerve cell. So you have neurons making connections inside your brain. And what's kind of cool, kind of metacognitive, is this idea that we're using our brains to study and consider the cells of our brains. So inside your brain right now there are cells that are reaching out and making connections with other cells, and all working together to allow you to even hear what I'm saying and understand what I'm saying and read your notes and go to class. So we're using our brain to study our brain, and we're using our cells to study our cells. And if you're thinking about that for too long, it kind of weirds you out. So I try not to think about it for too long. We're going to start right here with Concept 6.1. It says biologists use microscopes and the tools of biochemistry to study cells. And of course, in your lab activities, you probably use some microscopes so far. Probably the light type microscopes that you would normally use in a high school. Once you get up to the university level or the college level, you might have the opportunity to use an electron scanning microscope or some other kind of microscope that allows you to see further than a light microscope. But all of these tools are useful because most of what we're going to be looking at as far as these cells and the cell organelles are very complex, but they're far too small to see with our unaided naked eye. So as science has developed, as science has gone on, one of the things we've done is also we've been able to develop and further refine the tools that we use, including microscopes. Way back in the 1600s, some of the first crude microscopes were created, and you may have heard of some of these names. A little bit of history here. Robert Hooke was the first to actually document the scene of cell walls. He was looking at a sample from a cork tree, and what he saw in his crude microscope back in 1665 reminded him of the very sparse rooms that monks live in at monasteries. And that a word for that was cell. Of course, nowadays when we think of that type of cell, we usually think of a jail cell or a prison cell. But he was actually thinking of the tiny little dorm rooms that monks lived in a monastery, very sparse, very open, not much in them except for the four walls. So when he looked into his microscope, he saw what he considered to be cells, and he actually coined that term, cell, to describe the structures he saw in the cork wood underneath his microscope. Then a little bit further on, we have in 1674 Anton van Leeuwenhoek. Anton van Leeuwenhoek is considered kind of the father of microorganisms. He was the first to see actual living, moving microorganisms in his microscope. He called them little animalcules. And so Hooke and van Leeuwenhoek were kind of contemporaries, and they developed a lot of the microscopy technology that first began this revolution of seeing into this hidden world of microorganisms and tiny cells. The most common kind of microscope that we're going to use at the high school level is going to be a light microscope. So a light microscope uses visible light, and it passes it through a specimen and then through a series of glass lenses before it gets to your eye. And the lenses bend the light, they refract the light so that the image is magnified. So that as the light passes through the specimen and then through the lens, it gets flared out and magnified so that the object appears to be quite a bit larger than it actually is. Down here at the bottom of this slide, we have kind of an old picture. Obviously, these are hand-drawn. This is from Robert Hooke's very interesting book that he wrote called Micrographia, where he showed pictures of different specimens. Here are the cork cells that he was looking at, and he also had drawings and pictures of insects and other small little creatures, and this book kind of blew everybody away when it came out back in the 1600s, because this was kind of like a hidden world that no one had access to prior to the development of these microscopes. For any type of microscope that you look at, there's kind of three important parameters or three important pieces of information about that particular microscope. One of those is magnification, and that one is probably familiar to you. That's basically just how large can you make the object look. In other words, the ratio of the object's image size to its actual size. And so the more expensive the microscope, the larger the microscope, the greater the magnification that you can achieve. The next one is resolution. This one's also very important because resolution is the measure of clarity of an image, or what is the minimal distance between 2 points that you can distinguish. And so if you have, for example, in TV screens and computer screens, you've probably heard the term resolution, and it usually indicates a more massive amount of pixels or dots per inch. So the higher the resolution, the greater image clarity you can make out. And then the last one is contrast. And contrast is the difference in the parts of the sample. In other words, between the light and the dark areas. Sometimes if you don't have very good contrast, it's kind of muddy looking and it's difficult to distinguish between one area and the next area. So high contrast helps you visibly see the difference between one area of a sample and another area of a sample. Your textbook has this kind of cool chart that kind of shows you from the human size all the way down to the atoms what can certain microscopes or the unaided eye see. And so you can see that with your own eye you can see down to a certain level. Like you could see down to perhaps the size of frog eggs, slightly below 1 centimeter in length. Once you get past that, the unaided eye has trouble seeing things smaller, so you can see where a light microscope can get you down to a certain level. Human eggs, a lot of your plant and animal cells, even down into oftentimes some of your bacteria cells and things of that nature. Once you get smaller than that, even a light microscope has trouble, and the reason for that we'll talk about in just a second. But you can use an electron microscope of some kind, either a scanning electron microscope or a tunneling electron microscope, and you can get down even smaller. Now, can we see actual single molecules and atoms with a microscope? Not really. We have other ways of viewing that material, but it wouldn't be like a direct view of the actual object using a microscope. Light microscopes like you would have at the high school level the best ones can magnify about 1,000 times. So you can take an object and enlarge it about 1,000 times, which is pretty good. It gets you down to see some of the very small things. There are a variety of new techniques that we can now use with light microscopes to kind of enhance their ability to see. And of course, you can stain your sample or mark your sample with fluorescent dyes and things of that nature. That'll even help further make certain things stand out and be able to see certain details. Most of our cell parts that we're going to talk about are too small to be seen with a light microscope. So most of the organelles, like a mitochondria or some of the parts of the cell, might be too small to see for just a normal everyday light microscope. And the reason for that is actually kind of interesting because light waves themselves have a certain size, and if you're trying to look at an object that is smaller than the light wave is itself, then you're not going to be able to resolve that. You're not going to be able to see that using light. So for an example, let's say that you were bouncing tennis balls off of an object, and the tennis balls would represent light waves. Well, if the object, say, were the size of a car, you could close your eyes and you could toss a tennis ball back and forth as you moved from left to right, and you could kind of get an outline in your mind of that car from how the tennis ball bounced back to you. So if you're using an object that is smaller than the object you're measuring, you can kind of get an idea of what that object looks like. But once you get down to a very small object like the cell organelles, then the light waves are actually too large to get a good measurement. That would be like taking a tennis ball and trying to measure something very tiny with. That would be like trying to get a reading off of a thumbtack by using a tennis ball. Obviously, the tennis ball is too large and you wouldn't be able to bounce off of it and get any useful information. So the reason that light microscopes can't be used for the tiniest of objects is because the light waves themselves are actually too large. In your textbook, they do a great job of showing different types of microscopy and different variations of methods used. Here is an unstained specimen. If you're going to be looking at live cells, generally you're looking at unstained specimens. And the problem is that most of these cells are kind of clear and they're very difficult to see. So you can see in the picture the cells, but you notice that it's difficult to make out too much detail. Another way to look at the cells is to stain them, and that works very well to kind of show more contrast and more detail. But the problem is usually to stain a specimen, it has to be fixed and preserved, and that means the cells usually are no longer alive. But it does give you quite a bit more detail. Here we have what's called phase contrast. This is basically an optical modification that you can use with a microscope. Again, this works very well for unstained living cells. It gives you a little bit better view, but you're just modifying the light as it passes through to give you a bit more contrast. So this is not staining, it's just modifying the light as it passes through the microscope, and it gives you a bit more detail. Here you have another optical modification to enhance the differences in density of a material. So this is a different type of contrast technique where, again, it works very well for living cells because you're not staining them or affecting them in any way. So you can see and look at the living cells, but it gives you a bit more detail, kind of gives it a nice 3D effect. We do have some more recent advances in light microscopy that kind of make it a little bit easier to see detail, especially at the lowest level that a light microscope can see. We can use different fluorescent dyes that will attach themselves to different molecules and show up in different ways. We can use what's called confocal microscopy and deconvolution, and these are just ways to try to sharpen the image and allow us to see more than we could normally see. And then we have new techniques for labeling the cells to try to stain them or mark them in some way to improve resolution. In your book, they show some good examples. Here is using fluorescent tags or fluorescent dyes to mark parts of the cell. Here's an example of the confocal technique where you have at the top kind of a mixture of light that is in focus and out of focus, and then you can use a technique to try to get rid of the out of focus light and you get a more precise or cleaner picture down here at the bottom. And then deconvolution, again, is using another image processing software like a computer image processing software to enhance that image and clarify that image just a little bit further. Finally, we have what's called super resolution techniques, and this is actually using a combination of fluorescent dye and, again, light manipulation to further enhance what we can see and extend that light microscope down to the smallest levels. Once you get down to the size that light microscopy just doesn't work for you anymore, the next option is to use what's called an electron microscope. Electron microscopes are very good for studying subcellular structures, and the way they work is by basically, instead of shooting beams of light, you are shooting beams of electrons. And of course, electrons are very, very, very tiny, so you have, again, a finer ability to look at smaller objects. There's basically two different kinds. You have a scanning electron microscope, which is very good for looking at the surface of a specimen, gives you a nice, cool 3D picture, and I'm sure you've seen lots of these before. And then the other one is a tunneling or transmission electron microscope. A transmission or tunneling electron microscope actually passes electron beams through a specimen, and this would be like a specimen that you have sliced very thin and you want to look at the internal structure. Here's an example of a scanning electron microscope picture. This is cilia like you might find, say, lining your esophagus, or lining the bronchial tubes going down into your lungs, or perhaps within your digestive system. Down here is a cool link. If you want to click on this link, it takes you to some awesome SEM pictures of insects and other materials. And again, these are very popular because you see almost like a hidden world, and you get to see a nice 3D image. I actually did some of this work as part of my master's degree program. So we had to choose certain objects to look at, and I chose plants and plant pollen because it was springtime in East Texas and I was suffering from allergies. So these are some of the samples that I actually prepared and put underneath the scanning electron microscope at Stephen F. Austin State University. Up here at the top, we have some oak material. This is from an oak plant. This is an oak anther, which would contain some of the pollen. Here to the right, we have the actual male pine cone, and these little figures that you see that kind of look like Mickey Mouse creatures, these are actually the pollen grains of a pine tree. And then down here at the bottom, this is actually the male portion of a azalea flower, and this is actually the pollen of the azalea plant spilling out here. So these are all pictures that were taken with a scanning electron microscope. You'll notice they're in black and white. The scanning electron microscope only takes photographs or shows you images in black and white, but oftentimes they're colorized when you see them in print. So when you go to that link in the previous slide, you'll see that they have been colorized. That color is added via computer after the fact. The other type of electron microscope is a transmission or tunneling electron microscope. This is actually using a very thin cut sample, and it is sending that beam of electrons through the sample as opposed to bouncing them off of the outside of the sample. And this is primarily to get a look at the internal structure. So where we were looking at the external cilia in the previous picture, like right here, there's like an outside view of the cilia, kind of a nice 3D image. This would be looking at a cross-section of the cilia. So now you have chopped the cilia into a thin slice or section, and you're looking at the inside. Another thing that scientists do when we want to examine cells is we want to break those parts out. So we want to look at the different organelles, and the way we do that is by a method called cell fractionation. Cell fractionation actually takes the cell apart and then separates all the major organelles so that you can look at them kind of individually or separately. And it kind of sounds odd, but basically what we're doing is we're making a cell milkshake, and then we're spinning that milkshake at a very high speed to separate out the constituents or the separate parts. And this allows us to say if we want to only look at and study the mitochondria, we have a way to do that. Or if we want to look at and study the nucleus of the cell, well, we can separate the nucleus from the rest of the cell, and we can do various experiments based on that. And also, we have the ability to look at those individual parts and then further develop our understanding of how their form and function interact with each other. The basic idea in cell fractionation is to take a cell sample, like here on the left we have a number of tissue cells in our sample. We're actually, they call it homogenization, but basically what they're doing is they're just putting it in a blender and blending it until it's a frothy mix of cell parts. So instead of individual cells, now we just have this goop that's all of the various cell parts all blended together. And then they put it in a centrifuge. And a centrifuge is basically just a spinning device that you can see, like we have the cell samples in here. And the centrifuge is going to spin at an extremely high rate. And so what's going to happen is the more massive particles are going to be thrown to the outside and are going to collect in the very bottom of the tube. So you can actually do this a couple of different ways. You can do this actually a couple of different times as well. So you can run the centrifuge at a certain G load, basically, a number of times the force of gravity. And then you'll get the largest of the materials collected down at the bottom. That can then be poured off into the next one. You can increase the G load, pour that off into the next one, increase the G load, pour that off into the next one, increase the G load. So what you end up happening is you collect the heaviest objects at the bottom, you pour off the excess, then the next heaviest objects collect at the bottom, you pour off the excess, the next heaviest objects, and then down the line. You can see like the nuclei and some of the cellular debris, which are the largest, would collect first, and then you might get mitochondria and chloroplasts second. Then you might get some of the smaller organelles, and then some of the very smallest organelles would be at the very end, and that would be, say, your ribosomes. To get those down to the bottom, you have to spin this at 150,000 times the force of gravity. That tells you that these objects must be very, very small. Alright, we're going to go ahead and stop right there. We're going to try to cut this up and keep it in short little segments. Just to run back to these SEM photos because I love them so much. I had a great time in this class. This was actually an entire course about how to prepare specimens and take pictures using the scanning electron microscope. The cool thing about these samples is that you have to prepare them. You use microscopes to prepare them, of course. You dry them out. Then you actually coat them with a combination of gold and other heavy metals. And so that's kind of cool. There's a device that you use that puts a very, very, very fine coat of gold and other heavy metals on the specimen. And then you actually put them into the scanning electron microscope, and the scanning electron microscope is like an electron gun that fires electrons at your sample, and as they bounce off, the microscope takes a reading, and you get this information back in the form of a picture. Beautiful, 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 cool pictures. So if you get the chance, go to this link right here, and you can see some other amazing photos taken with scanning electron microscopes. We'll take a break right here, and then we'll pick it up. Thanks, guys.